Okay, so what I'm going to show you is the fungal prep part of the lab. So what we're going to do is basically inoculate 2 plates with fungus. We'll inoculate 1 plate with Ficomyces, the other plate with Swordaria Prep. Once we inoculate those, then we'll allow them to grow. For a week, it takes about a week of incubation. Fungi incubate a lot longer than bacteria do. Usually bacteria is 48 hours, fungi are a week. And then after that week, we will then look at the plates and examine them and look at the mating in the fungi. Okay, so now I'm going to start with the Ficomyces, which are these 2. I have a plus strain and a minus strain. I'm going to put that on potato dextrose agar. We'll put the plus strain on one side, the minus strain on the other. So I'll go ahead and label this plus over here and minus over here, so we make sure that we know which strain goes on which side. Now I'm going to light the Bunsen burner. We'll turn this on until we barely hear it. Get a nice flame going. And I'm going to use tweezers and a scalpel to help me out here. So we'll basically flame that scalpel there. Okay, we'll flame those tweezers because we want to make sure we have a sterile technique. We'll open this up, cut a little square out of this guy here. Okay, this is the plus, so then we'll put the plus over here. Sometimes it takes tweezers to get that off of the scalpel there. Okay, I'll cover the lid really fast. We'll flame this so we get rid of all the plus strains over here. Move this guy out of the way. We'll then get the minus strain. We'll cut a square out of this minus strain. All right, this one has a lot more hyphae in there. And we will put the minus strain here on this side of the plate, if we can. Okay, lid on. We'll flame these guys really fast. Lid on, and our Ficomyces is ready to go. Okay, next then we want to do our Swordaria plate. So we have a tan strain of Swordaria. So Swordaria Prep tan, you can see how tan that coloration is. And we have a wild type strain, you can see how dark that one is. So this one's a lot darker than that one. In this case, we're going to just flame these and put these down. In this case, we're going to put a tan on one side, a tan on the other side. Then we'll put a wild type on one side and a wild type on the other side. So we're actually going to do 4 on this one because it works a little bit better with 4 than with 2. Okay, so we'll flame. Make sure we have none of that Ficomyces on these things. It's going to sizzle a little bit. Shh. We'll cool that one off too. Okay. Now we will go here and just cut out. In this case, I'm going to cut out 2 squares instead of 1. So then we'll put one on this side. Put our second one on this side. And we want to set it down. Because I dropped that, we're going to get some hyphae growing there, which should make an interesting plate later on. But it'll be fun to look at. And now we'll flame it. Make sure we kill all of that tan strain. Then we'll go over here and we'll do our wild type strain. Okay, again, we're going to do a couple of them. So I'm going to cut that, and then we'll cut it along here. And we'll just scoop that puppy out of there. There we go. Then we'll scoop that puppy out of there. And there we go. And reflame these guys, make sure we sterilize them. And finally, close the lid. Now what I'm going to do is I'm going to incubate these. So these guys I'm going to put in the about 28-degree incubator. These guys actually require even a little bit cooler than room temperature, so I'm going to stick them up in the windowsill so it's a teeny bit cooler for these guys to make.